here before we move it on. Okay, so the next topic that I want to do is basically over 2 hours. So we'll start this today and we'll finish it off tomorrow. And this is cytogenetics. Lots of stuff in cytogenetics. And so it does really require a couple of hours to go through all of this material. And so the objectives at the very beginning of this are for both hours. And so you're going to see quite a few objectives, but since it's for 2, it's hopefully not an overly excessive amount. But there's a lot of stuff with respect to cytogenetics. And I'm not going to go through all of the objectives. Recommended reading, Medical Genetics and Thompson and Thompson. Either one of these would be good to take a look at. So we'll start very early on. Prior to the early 1950s, which is about when we first figured out the double helix, we weren't sure how many chromosomes humans had. You know, think of the progress from 1950 to now, where we're talking about sequencing 100,000 genomes. In 1950, we didn't even know how many chromosomes we had. The consensus at the time was 48. Hopefully everybody says that's wrong. We have 46 chromosomes. So the consensus was wrong in terms of the number of chromosomes. So why would we not know? And a major reason why is that techniques to visualize chromosomes were very poor. Another thing is you have to look at a cell at just the right period of time, which essentially is M phase, when the chromosomes are condensed. So if you looked at, for instance, a typical hepatocyte in your liver, how often does that hepatocyte go through the cell cycle? In other words, complete a cell division. It's about once a year. Once a year, a typical hepatocyte divides. So if you are isolating a biopsy from the liver and looking for mitotic cells, you are probably not going to find any. If you are looking at interphase, the chromosomes are so extended that you can't tell one chromosome from another. And so the techniques for seeing cells at the right time were very poor. Thus, we didn't have a good understanding of chromosomes very much at all. From the 1950s, there were a number of techniques that I'm going to talk about that really revolutionized this type of analysis and really improved our abilities. So, one was the use of spindle poisons, colchicine and colsumid. Some of the most dangerous compounds that you could mess around with, to tell you the truth. These destabilize microtubules. Microtubules are involved in the migration of chromosomes during M phase. So the separation of chromosomes. So if we destabilize the cells, or destabilize the microtubules, the cell gets stuck at metaphase and cannot go beyond metaphase. So it represents a block in terms of the cell. So they will accumulate then at that particular phase. Another is to isolate these cells and incubate them in a hypotonic solution. A solution that is low for potassium chloride. That causes water to rush into the cell and separates the chromosomes. Good question here. If you are looking at cells that are in M phase, essentially going through mitosis, is there a nuclear membrane present? And hopefully, no, there is not. The nuclear membrane breaks down. So if you then do a hypotonic solution, the cell swells, these chromosomes will essentially spread out through the entire cell because there is no nucleus any longer. So they can occupy the entire space that cell has. And then staining techniques that allow for identification of the individual chromosomes. So how do we do this? The first thing we have to do is collect tissue. And the question is, what kind of cell do we want to look at? A lot of the time, it's just white blood cells. If we're just looking in general, we isolate white blood cells. If we're looking at a specific cancer, If it's a leukemia, we could still isolate white blood cells. If it's a solid tumor, we would have to do a biopsy and isolate those particular cells. But oftentimes, it is white blood cells. We take those cells, and one of the things we want to do is to stimulate their proliferation. So we grow them in culture for about 48 to 72 hours. This allows for these cells to divide, to proliferate. The white blood cells, the lymphocytes, are what we're looking at. Red blood cells are of no value to us because they don't have a nucleus. So we add a compound called phytohemagglutinin. And phytohemagglutinin does a couple of things. One is it is a mitogen for lymphocytes. So it stimulates those lymphocytes to go through the cell cycle. It also clumps the red blood cells. So the red blood cells then fall to the bottom of the tube. And so we can nicely get rid of the red blood cells and stimulate the lymphocytes to divide. Because if we don't stimulate them, we're just not going to see cells in mitosis. So after either 48 hours or 72 hours, a cell cycle for a lymphocyte is about 24 hours. And so we stimulate these cells at, say, 10 a.m. Then we come back 10 a.m. 48 hours later, or 10 a.m. 72 hours later, and a large number of the cells should be at the phase that we want, which is mitosis. So we add the coulsomid. That's going to induce the metaphase arrest. We're then going to isolate these cells and then incubate them in the hypotonic KCl solution. That's going to cause them to swell and become very fragile. We don't want them to lyse, though. So the concentration of KCl has to be just right. Those cells swell, and after a certain amount of time, we take those swollen cells very carefully and we drip them onto a nice clean slide, glass slide. When those cells with this turgor hit the glass slide, they burst open. The chromosomes have a very high affinity for glass. So when they burst, the chromosomes spread out onto the glass slide, and they will stick and they will adhere to the glass slide at that point when the cell hit. This would be an example. This is a cell that was in interphase, and the interphase cells tend not to swell as much, and so they tend not to burst. But then this is a cell that did burst, and then we can do staining. I give you this picture because this is actually me. I did this when I was a graduate student and kept the picture for all of these different years. And so if you count these, and I encourage you to do so, you will find that there are 46 in there. So I was greatly relieved to find that I did have, in fact, 46 chromosomes. If you just look at the glass slide, you wouldn't be able to see the chromosomes at all. We have to stain the chromosomes to make them visible. Chromosomes are not naturally dark like this. So we have to do staining. So spreads and karyotypes. This represents a spread. So it has hit the slide, the chromosomes have spread out, and then we have stained them. We view them under the light microscope and photograph the best ones. And all of this is done digitally now. And it's very much computer aided. But essentially, these people are amazing, the cytogeneticists, in terms of their abilities to take these chromosomes and identify which one is which. And you would be very impressed. I highly recommend it if you ever have a chance to go to a cytogenetics lab and see what they do. And you'll see technicians who are sitting down with computer programs where they can take a spread and they can click on the various chromosomes and move them around and basically create a karyotype that way. It's very, very neat to see. And so if we look at this particular spread, this is from an individual who is trisomy 21. And this was after what's called G-banding. I'll describe G-banding in a second. But here's one of the chromosome 21s, here's a second chromosome 21, and here's a third copy of chromosome 21. And so a diagnosis of trisomy 21 comes from this type of cytogenetic analysis. And you usually have to identify at least 10, between 10 and 20 spreads that show the same karyotype to be able to make that diagnosis. And so that's what the cytogeneticists are going to be doing. This is a spread as opposed to a karyotype. So what we have to do next is arrange the chromosomes in a specific order, largely based on their size. And that's the actual karyotype. So this is what you're going to see then when you order a karyotype for an individual. We go from chromosome 21 chromosome 1 down to chromosome 22, and then the sex chromosomes are placed in the lower right corner all the time. And so you can look at this and say, this is an individual who is a normal male. You may not be able to tell that just by looking at it, but you can certainly tell that it's a male because of XY. So we arrange them, largest to smallest. We have 2 copies of each and every chromosome. You have a maternal chromosome 1, a paternal chromosome 1. Can you tell? Not here. Notice that the chromosomes are not always perfectly straight either. They vary depending upon how they adhere to the glass. So there's nothing wrong with this chromosome, it just happens to have adhered in this way. What about something like that? Or there's actually 2 of them on here. And there's this one. They look like strange regions of the chromosomes. What this actually is when this chromosome spread occurred, there was overlap between 2 chromosomes. So there's actually another chromosome that was laying on top of this one. And so essentially what they did was then they took 2 pictures of this particular spread. And in one picture they looked at this chromosome, and another picture they looked at this chromosome, and another different picture they maybe looked at that chromosome. So that does not represent like something funky about the chromosome itself. You can see another one here. Just that there was not a perfect spread in all of the chromosomes. And so nothing to be alarmed, this is a normal chromosome spread. We organize these into particular groups, and we label those as A through G. And usually the labeling is not shown on a karyotype, but the organization is maintained. And so there is an A group that consists of chromosomes 1, 2, and 3. And so that's placed up in the upper left. Then you see that there's a space. This represents the B group, 4 and 5. Then you see a large group 6 through 12. That represents C. D is 13, 14, 15. Then E is 16, 17, 18. F is 19 and 20 down here. Then a space, then 21 and 22 after that. I'm not going to require you to know which chromosomes are in which groups at all. All you should know is that this is the basis for how they provide a karyotype. So it's very common to see this space here, these all in a single row because they're all grouped together. And I'll describe these differences in these chromosomes as we work through this. So we classify these first by size. And so the largest chromosome is chromosome 1, and then they go down from there. Chromosome 21 is actually the smallest human chromosome. But it's not the smallest number, or largest number, if you want to call it that. They made a mistake. Didn't know the total size of these chromosomes. This was done when they were first developing these techniques. Cytogeneticists got together at a convention in Paris in like the early 60s or so. And they said, we have to standardize this in terms of how we're going to do this, because everyone is now speaking a different language with respect to chromosomes and such. And so at this Paris convention, they laid down certain rules. And one was the designation for each and every chromosome. So chromosome 21, then this is chromosome 22, they said. So this chromosome is actually a little bit smaller, chromosome 21. After size, they are classified according to the location of the centromere. And we can basically categorize 3 types of chromosomes. First one is metacentric. And metacentric chromosomes are basically divided into 2 equal parts for where the centromere is. So it occurs near the middle of the chromosome. It's a metacentric chromosome. Then there are acrocentric chromosomes. Those are where the centromere is very close to the telomere on one of the 2 arms. And then there are submetacentric chromosomes, which kind of split the difference between these 2. It's not at the midline, but it's not super close to the telomere. And so if we give examples, chromosome 2 is a metacentric chromosome. You can see it has the upper arms. This constriction represents the centromere. And then this is the lower arm, and they are about the same length. This one is a submetacentric, and so it has a significant shorter arm, which is called the P-arm, which I'll describe in a second. and then a much longer lower arm or arms. And then we have the acrocentrics. The acrocentrics is where the upper arms are present but extremely small. One other point that I want to make with respect to the acrocentric chromosomes is the P arms of the acrocentric chromosomes are where the ribosomal RNA genes reside. So there are a number of acrocentric chromosomes. There are 10 per diploid karyotype. And in each one of those, the ribosomal RNA genes reside within the P arms of those chromosomes. A small point, but kind of an interesting one. So, P arms and Q arms. At this same Paris convention, they gave this designation as well. The short arm of the chromosome is oriented to the top of the chromosome, and it is designated the P arm. And because it was in France, P was for petit. And so short arm, P. Then the long arm is, let's say, what do we call that? Well, if we're going to call it P, let's call the next one Q. Just because P and Q follows P, simple enough. So the long arm is the Q arm. It is always oriented to the bottom of the chromosome. So we have the P arms and the Q arms of the chromosomes. If it's a metacentric chromosome, the P arms and the Q arms have largely been assigned. And really, it was based upon detailed analysis. No human chromosome is truly, absolutely metacentric, meaning that it's a perfect break. And so there is a long arm and a short arm, even on a metacentric chromosome. And so there is a P arm and a Q arm for those as well. So chromosome 2 always has the same short arms oriented to the top and the long arms oriented toward the bottom of the chromosome. Now, the next thing to develop was banding techniques. These were really developed in the 1970s, and they were huge with respect to being able to identify each and every chromosome uniquely. How the staining works is not entirely clear, but there's a number of these different techniques. So here's some differences. There's what's called G banding, there's R banding, and then there's C banding. Those are 3, plus one other one that I want you to be familiar with. This one is just GIMSA staining. And GIMSA staining, if you just add GIMSA to chromosomes, it stains them black. And so that's what you see. Now, you see chromosome 1, 2, and 3. If you were to look at these two chromosomes, you'd probably have a very hard time saying which one is number 1 versus number 2, just based on GIMSA staining. So GIMSA is what we had to start with. But it stained the entire chromosome black. Then G banding came along, then R banding, and C banding. So I'll talk a little bit about each of those methods. First, G banding. The G in G banding stands for GIMSA. And essentially, this is the most common staining technique for chromosomes you're going to see. So the vast majority is G banding. And if you see ideograms, which are basically cartoon representations of chromosomes, they're going to show you G banding for this. And essentially in this technique, we use GIMSA stain, and we also do a partial digestion of the chromosome with an enzyme called trypsin. And trypsin will differentially kind of loosen up different regions of the chromosome. And so if it loosens up the region, it takes up the stain. Whereas if the region stays tight, it doesn't take up the stain nearly as well. And so with GIMSA staining, or with G banding, sorry, what we see is alternating light and dark bands on the chromosomes. And so when you see something like this and you see the light bands and the dark bands, that is due to G banding. Then Q banding. Q banding is not here, and the fundamental difference with Q banding is its fluorescence. You use quinacridone, and that's the basis for the Q. Quinacridone is a fluorescent dye that acts very similarly to what GIMSA does, except that it will be fluorescent. You see the same G banding pattern if you use trypsin, but you have to have a fluorescent microscope in order to do Q banding. So most labs do G banding. Very few do Q banding, but it's possible. R banding, the R stands for reverse banding. With reverse banding, there is a heat treatment that is part of the protocol. And for some reason that nobody understands, if you do this heat treatment, it actually reverses the staining pattern. So what was a light band with respect to G banding becomes a dark band with respect to C banding. So you notice on this chromosome 1, if you look at the telomeric region, it is very, very light, and so it's hard to see. Whereas with R banding, it stains very heavily in that region. This is useful if you're looking for alterations to the very ends of the chromosomes, because the ends of the chromosomes tend to be light, and so they tend to be difficult to see exactly where the chromosome ends with G banding. With R banding, it's much easier to detect that. And then C banding, the C stands for centromere. And with C banding, what we are staining for is highly repetitive DNA. And centromeres consist of highly repetitive DNA. And so if we look at chromosome 1 with C banding, that's where the banding is preferentially localized. And then chromosome 2, again, we see the centromere. Chromosome 3, again, we see the centromere. Not entirely. You'll notice that there is highly repetitive DNA in other regions, but the centromere really lights up. So if we are looking for an alteration that might involve the centromere of a chromosome, we would perhaps do C banding to be able to detect that. The last one that I want to talk about is what's called high-resolution banding. So high-resolution banding is usually G banding, but we isolate the chromosomes earlier in the process, in prophase or prometaphase as opposed to metaphase. In metaphase, the chromosomes are much more constricted. So here's a mid-metaphase chromosome, and here would be a prophase chromosome. The prophase chromosome is much more extended, and so there's a lot more information that we can potentially glean from high-resolution banding. The number of bands visible goes from about 400 if you are looking at these chromosomes to about 800 if you're looking at these chromosomes in prometaphase or prophase. And so we can see more detailed, more subtle changes in the chromosomes if we're looking at these extended chromosomes. As you can imagine, this technique is harder to do. If the chromosomes are more extended, they tend to overlap a whole lot more when you do the spread. And so this is a technique that requires a lot of specialization, and it's really only ordered if it is absolutely necessary. So if it's a really subtle abnormality. Largely another process called FISH has replaced this. So, band identification. Each and every visible band on the chromosome is given a unique identification. And you should have an understanding of this identification process. So we break down each chromosome into first the P arm and the Q arm. That's the first step. Is it the P arm or is it the Q arm? Well, the first step would be what chromosome is it? So chromosome 1, then P arm and Q arm. Then the P arm is broken down into regions and the Q arm is broken down into regions. The same Paris Convention basically gave us this designation as well. Another thing about regions is everything originates at the centromere and radiates outward toward the telomere. So region 1 on the P arm is closest to the centromere. Region 1 on the Q arm is closest to the centromere. Then it goes region 2, then it goes region 3. And then on the Q arm, region 1, region 2, region 3, region 4. Why does the Q arm have 4 regions and the P arm 3? That's what they decided to do. It is entirely arbitrary in terms of how they decided. What is the break point designation between region 1 and region 2? Not going to require you to know that kind of detail with respect to these chromosomes. Just the idea that you break it down that way. Then within a given region, say here's region 2. So within region 2 we have band 1, band 2, band 3, band 4, band 5. So there are 5 bands within region 2 on the Q arm of chromosome 1. Both light and dark bands are identified as bands. People often think just the dark bands are bands, but the light bands represent bands as well. So the first one is here, second one here, light band, then dark band, then light band. And so all of them are identified. So the numbering also starts from the centromere and extends toward the telomere. So it's going to go 1, 2, 3, 4, 5 as we go down. And here, 1, 2 as we go up. So 1, 2, 3, 4, 5, 6 going upward on that chromosome. So if you said the 5th band in region 2 of the Q arm of chromosome 1, those are the words. But you can simply designate that as 1q25. This is what you want to be familiar with in terms of what this means. This means chromosome 1, Q arm, region 2, band 5. And so then we could go chromosome 1, Q, region 2, band 5, right there. So that's how we identify gene locations as well. So if we said a particular gene is at 1q25, you could specify roughly where that is. Okay. Make sense? You certainly don't have to memorize. Thank you. You don't have to memorize these locations. Some of them will become automatic, like CFTR is 7q31.2. I just know that. It will never leave my brain. So we often do this in schematic form as well. So you can see lots of different schematics for the chromosomes. One thing to note is that, notice here's chromosome 1, and here's chromosome 1. On this one, they're showing 4 distinct arms, 2 P arms and 2 Q arms. Over here, they're showing 1 distinct P arm and 1 distinct Q arm. When you typically do this kind of staining, the P arms of the chromosomes tend to stick together during this staining. So you can't identify distinct P arms and Q arms. They are there, but they're held together by a protein called cohesin. So you don't really see this. This is more realistic. But when you are looking at this drawing, it's important to realize you are looking at what are called 2 sister chromatids per homolog. So there are 4 arms on this chromosome, even though you can't see it. And there's 4 arms on this one and 4 arms on this one, 2 P arms and 2 Q arms. So this is more realistic in terms of what the chromosome kind of looks like, but this is more realistic in terms of on a karyotype, it's what it looks like. That causes confusion for students because they don't see the arms. The arms are there, they're just glued together with a protein called cohesin holding them together. So if I give you a band identification, you should be able to roughly localize it to the part of the chromosome where it resides. Now, I'm going to give you something where I expect you to know that here is region 2 as opposed to region 3 or something. Absolutely not. But you should be able to get ballpark range. Like if I give you 1q25 and you point to chromosome 22, sorry, you got that wrong. Now, the other thing to note here on this figure is it's showing how many base pair there are on each and every one of these chromosomes. So chromosome 17, for instance, 88 million base pair. Just to give you a sense for how large these chromosomes are. And then the last one, here's chromosome 21, 45 million base pair, chromosome 22, slightly larger at 48 million base pair. And then the Y chromosome, 51 million base pair. And then this also represents the pattern that you would see with G-banding as well. So high resolution, when you do high resolution, you go from 400 bands to 800 bands. And so we get additional bands. These are what are called then subbands on the chromosomes. So large dark regions tend to be broken up. Like for instance here, there's one big band. But if you do high resolution banding, it breaks it up into multiple dark bands with light bands in between. So we call these subbands, and they are designated with decimal points. So 1p31 is a large dark band, that one up there. So if you get multiple new bands, it's 1p31.1, 1p31.2, 1p31.3, so on and so forth. So when I said earlier CFTR is 7q31.2, that has been localized with high resolution banding in terms of its location. So that is subband 2 of 31.2. Okay. Make sense? So far so good. Okay. So nomenclature. Got 1 minute left. I'll do this slide and then we'll wrap it up. Normal female is 46, usually comma, although I didn't add it, 46XX. Normal male, 46XY. If there is an abnormality in the chromosomes, it's usually after that designation. So we always start with the number of chromosomes that are present, then the constitution of the sex chromosomes. If it is a change in chromosome number, we note that. So if it involves a particular region of a chromosome, it's noted as well. So if we said we have diagnosed a newborn with trisomy 21 Down syndrome, that specific designation for this individual would be 47XX plus 21. In other words, we have 47 chromosomes instead of 46. It is a female, and the extra chromosome is an entire copy of 21. And then the karyotype for this individual would be this. And what you'd be looking for then is down here. 1, 2, 3 chromosome 21s, as opposed to 2. So I could certainly give you a karyotype like this and say, what is the most likely diagnosis? And you would need to be able to pick out what is the abnormality. Hopefully here it's pretty straightforward that you have an extra copy of chromosome 21. Am I going to eliminate these numbers so you'd have to know this is 21 based on staining? No, nothing like that. So just to be able to look at this and say, okay, there should be 2 copies of every one of them. There's 3 copies here. What is that? Down syndrome. Okay, so when we come back after a break, we will take a look at this figure, and I will scare the hell out of you with it.